Hi guys, today I'm going to be talking to you about immunology, something that people seem to get very confused about, so I'm going to go through it with you nice and slowly and carefully. Right, so all around us, on surfaces, on our bodies, in our food, there are bacteria, viruses and fungi. Most of these are harmless, but some of them can be harmful to us, and these are called pathogens. Pathogens like to reproduce inside human bodies or on our skin, and they can cause infections and make us poorly. So these critters are constantly trying to get into our bodies, but luckily we have some defenses. The first line of defense we call barrier defenses, and these are things like your skin, which helps stop bacteria and viruses and fungi getting in, enzymes in your tears and your snot and in your ears, which contain lysozyme enzymes, can break down things trying to get into your eyes, break down the membranes of bacteria and fungi. The pH in your stomach and also in your genitalia can stop pathogens getting in that way. And you've also got cilia in your lungs, on your lung cells, to help expel anything that you may breathe in. Okay? Now this might be a good time to remind you guys that all cells have receptor molecules on their surfaces. Like this body cell here, you can see it's got little receptors on the outside, and so is this foreign pathogen here. Now this is how cells recognize each other, okay? If it's not recognized, it's called an antigen, okay? And these antigens here can cause an immune response in your body. The body will recognize them as foreign, and an immune response will be triggered. Now these can either be the antigen receptors on the surface of the bacteria, or the toxins that some of these bacteria or viruses can produce. Okay, so if something manages to get into your body, we have another line of defense, which is also non-specific, which means it'll just attack anything it finds that's not human. And these are a specific type of white blood cell called phagocytes. These are easily recognizable by their huge nucleus. It takes up most of the cell. They've also got loads of lysozymes. Remember these from our organelle section, which contain hydrolytic enzymes to break stuff down. Right, so this phagocyte has encountered a pathogen. It may have been following a chemical trail, possibly the toxins it was producing that led it to it. And what happens is it binds to it, recognizes the receptors as foreign antigens, and engulfs it through a specialized type of endocytosis called phagocytosis. It encapsulates the pathogen in a vacuole. The vacuole binds to one of the hydrolytic enzymes, and they break it down. The lysosomes bind to it and break it down. Once it has been broken down into just basic proteins and carbohydrates, what the phagocyte does is move the antigens from the pathogen onto its own cell surface here. And we call this an antigen presenting cell, okay? APC for short. So it's presenting the antigens, which helps sort of stimulate some of the other immune responses, and it also releases some chemicals, okay? These chemicals trigger our specific immune response, okay? Now the first line of this response is to do with T-cells, which are another type of white blood cell. These lie dormant in your body all your life, and they can be activated by the chemicals set off by the phagocytes. So once they have been activated, they start dividing by mitosis into lots of different kinds of T cells. You don't really need to know all of these. The main ones you need to know are the memory cells and the killer cells. But I'll go through all of them because they're quite interesting anyway. So one of them is a T helper cell, which goes and activates B cells, which I'll talk about in a moment. The killer cell, also known as natural killer cell, NK for short, attacks any antigen-presenting cell, which is an infected body cell. So the cell's going, help me, I'm infected. And the natural killer cell will come along, binds to the cell and break down its membranes, thus sort of exploding it, lysing it, so that it's not alive anymore and it can't produce any new bacteria and viruses. It also produces memory cells, which stay within the body in the lymph system and remember this specific type of bacteria, so we can't get infected a second time. There's also these T suppressor cells, which you don't need to know about, but I think they're pretty interesting. They basically stop the whole process at the end of the infection. So there's got to be something that says, wait a minute, the infection's over, we can stop now. Okay. Now, going back to these T helper cells, they have activated these B cells, which are also specific and lay dormant most of the time. So they're activated by the helper cell. Once they are activated, they also start dividing into lots and lots of other B cells by mitosis. One of these is another memory cell, which memorizes, in a way, how to destroy this particular pathogen. And the other kind are called B plasma cells. Now these are very important because they make something called antibodies. Just a quick reminder about antibodies. They're quaternary proteins and they have two specific binding sites to bind to antigens, which means they can bind to two antigens at once. They look like little Ys, but they're made up of two protein tertiary subunits. Okay, so there are loads of different kinds of B cells, and each B cell has these different receptors on them. The body is very clever with this. It will find the receptor that is most likely to fit onto the pathogen's antigens. So it kind of is another shape and fit thing going on here, just like with most protein processes. It'll find the antibody receptor that fits best to the antigen and then start producing millions and millions and millions of them, okay? This takes quite a while, so you might feel ill for a couple of days and then slowly you'll start to produce hundreds and hundreds and hundreds of these antibodies, and they are very, very specific, or as specific as they can be with what B cells are available, and these start binding to the antigens on the pathogens, and this makes them stick together so they can't get into your cells anymore. And it also attracts phagocytes, which we know like to engulf and digest pathogens. Now let's try and represent this in a graph. So number 1 here on our graph shows that the primary infection, the first time you get infected with something, you've got a very slow production of antibodies, and you also don't produce many, okay? So maybe this is the first time you've had a specific type of cold virus. The second time we get infected, the body recognizes this pathogen because of our B memory cells and our T memory cells, so it can very quickly kill any cells that are infected and make antibodies to kill the pathogens themselves. And then 3 here, we've got another different type of pathogen, or possibly the same pathogen but with different antigens, like the flu virus, which can change its antigens. It can change its antigens sometimes periodically to reinfect the same host, which is why you need a flu shot every year, flu vaccination every year, because they just keep changing their antigens. Okay, so that's a nice summary of immunology, what you need to know for A-level. Hope it helped. Thanks for watching. Goodbye.